Okay, well, it's 20 past on my computer, so I'm very happy to take over and chair session number 2, which is a very busy one. And I'm very happy to introduce Sara, who is the driving force behind this meeting with all the EACR people, and helped a lot with Janine and Nita to organize this meeting. So she is actually coming from the Cancer Research UK Bitson Institute, and she had obtained her PhD in the lab of Guido Serini and Federico Bussolini in Italy, in Torino. And she then moved to the lab of Matthias Mann, right, until 2010, where she performed her postdoctoral work before actually establishing her team again at the Cancer Research UK Bitson Institute. And I'm very happy to launch this session number 2 and looking forward to your talk, Sara.
Thanks, Jackie. Thank you very much for the introduction. Okay, I'll share my. Okay, hello everyone. So just to tell you a bit more about my group. So we are very interested in working on aggressive cancers, in particular triple-negative breast cancer and high-grade serous ovarian cancer, because they have a very few therapeutic options, particularly to target the cancer cells. However, these tumors have a very large portion composed by stroma, as you can see from this picture. And this is an ovarian cancer metastasis in the omentum. And basically, we want to find the ways to exploit the stroma as an alternative therapeutic target. And within the stroma, we are particularly interested in cancer-associated fibroblasts, and as you may know, and we'll hear more about it during this meeting, there are different flavors of these cells. So these cells can have different functions, different phenotypes, and we are particularly interested in those fibroblasts that produce matrix, that are also called myofibroblasts like CAFs. And we want to understand how they influence the behavior and functions of cancer cells, immune cells, and endothelial cells. And we are particularly interested in targeting these cells because they are producing extracellular matrix. We want to inhibit the production of extracellular matrix. Today we have already heard how important it is in tumor progression and metastasis. And also my lab has contributed in the past to understand how CAFs remodel the extracellular matrix and how this can contribute to cancer. For example, by inducing the sprouting of blood vessels, or inducing cancer cells to intravasate and form a distant metastasis. We have also shown that they can remodel the matrix to stimulate the growth of the cancer cells and also their invasive behavior. And an interesting aspect that is a bit more new about the extracellular matrix is that it's also very important to alter the immune tumor microenvironment, for example, to induce a chronic activation of the T cells or exclude immune cells to access the tumor. And this is very important to basically make patients resistant to immunotherapy, for example. So we want to find the ways to target the production of extracellular matrix by CAFs, but how can we do that? And we are particularly focusing on the metabolism of cancer-associated fibroblasts, and this is for two reasons. First, because the metabolism can generate acetyl and methyl groups that can be used, for example, for epigenetic regulation by providing basically acetyl and methyl groups for post-translational modification, for example, of histones for methylation and acetylation. But also metabolism can produce amino acids that are very important for protein translation. And we know that when fibroblasts get activated, they really start producing a lot of different extracellular matrix proteins and other secreted factors, suggesting that maybe targeting the availability of amino acid can impact on this particular aspect of CAF transformation. And we studied the link between metastasis and extracellular matrix by using patient-derived fibroblasts. So the story that I decided to tell you today about is focused on breast cancer. And we isolate our CAFs either from breast cancer tumor tissues or from normal tumor adjacent tissue. And as you can see, here from this atomic force microscopy analysis of matrix produced by normal fibroblast or CAF in culture, you can see indeed that CAFs produce a much more matrix, that the structure is completely different. And when we quantify specifically the total amount of collagen, you can see that CAFs always make more collagen than their normal fibroblast counterpart. And we have done extensive mass spectrometry proteomic analysis of the matrix produced by CAF in culture. And what we have found is that just as 300 of proteins basically make more than 95% of the total extracellular matrix mass. And among the most abundant ones, we have collagen 1, collagen 6, and collagen 12. And we also found that these three collagens are also the most abundant ones found in xenografts. In this case, we have transplanted mice with MCF10DCIS.com cells and patient-derived CAFs. And here, basically, you can see that both the human CAFs and the endogenous stroma of the tumor both produce these three collagens very abundantly. And we know that also these three collagens are highly expressed in patient samples. Here we have analyzed the publicly available single-cell RNA-seq data of triple-negative breast cancer patient samples. And you can see, particularly if we look at these myofibroblasts like CAF that make a lot of matrix, that these three collagens are highly expressed. But with our proteomic analysis, we also found that more than 30% of the CAF-derived extracellular matrix is actually made by collagen. So, and why is that important? The reason is that the collagens are very unique proteins in terms of amino acidic composition. In fact, in collagen, you often have the repetition of tripeptides, and one of these tripeptides is composed by glycine, proline, and hydroxyproline. And this basically leads to make collagens containing huge amount of residues of glycine and proline when you compare them with other proteins in the rest of the protein. So they have this very unique abundance of amino acids, and the question is, can we then target the availability of specific amino acids to disrupt collagen production? And this is basically the theme of the story that I'm telling you today. And this story started quite a few years ago when we did a very extensive mass spectrometry-based molecular characterization of normal fibroblasts and CAFs. We basically measured the proteome, the phosphoproteome, the acetylome, and also we did quite a few tracing experiments to look at the metabolism of the cells, because we really wanted to understand what happens to the fibroblasts when they get activated. And I really wanted to highlight also, as you have seen in Janine and Tom's talk, the power of using these unbiased mass spectrometry-based approaches. Now, one of the first findings that we had is that in the CAFs, the pyruvate dehydrogenase was one of the most downregulated phosphorylation in our CAFs. Now, this phosphorylation is regulatory and is inhibiting the pyruvate dehydrogenase. So what we found is that in CAFs is less phosphorylated, and doing an enzymatic assay, we could indeed show that CAFs have a pyruvate dehydrogenase more active. Now, the pyruvate dehydrogenase is a mitochondrial enzyme that generates acetyl-CoA from pyruvate. And then the acetyl-CoA can either go through the TCA cycle, or it's converted into citrate. The citrate exits the mitochondria. then it can be converted back into acetyl-CoA by the ATP citrate synthase. And in this way, it can be involved in protein acetylation or lipid synthesis or the synthesis of ketone bodies. And what we found is that in our model, this acetyl-CoA, this increase in pyruvate dehydrogenase leads to an increase in acetyl-CoA production from glucose in our cancer-associated fibroblasts. And here you always see pairs of normal fibroblasts and CAFs coming from different patients. And when we look at the expression of pyruvate dehydrogenase kinases, which are the kinases that regulate the phosphorylation of the pyruvate dehydrogenase, what we found is that in CAFs, all the pyruvate dehydrogenase kinases, which are 4, are all expressed at very low levels. What we also found is that the most expressed one in the normal fibroblast is PDK2, suggesting that PDK2 may have a major role in regulating pyruvate dehydrogenase activity. And what we show here is that indeed this is the case, because when we overexpress the pyruvate dehydrogenase kinase in our CAFs, we induce an increase in phosphorylation in pyruvate dehydrogenase, and this corresponds in a significant decrease in acetyl-CoA production from glucose. Conversely, when we take our normal fibroblast, and then we silence PDK2 to mimic what happens in the cancer-associated fibroblast, this is sufficient to decrease the phosphorylation of the pyruvate dehydrogenase and to increase the production of acetyl-CoA from glucose. Another interesting observation from our omics analysis is that we found that CAFs have histones typically hyperacetylated. And those that I have highlighted here are lysines on histone HS3, and these are typically acetylated by the histone acetyltransferase EP300. So altogether, we found these results very intriguing. And this because there are several reports showing that the levels of acetyl-CoA can basically regulate the level of acetylation of histones, including HS3, and that these can occur via activation of EP300. What is also very interesting is that histone 3 acetylation, including HS3K27, is a hallmark of gene expression activation, and it has also been found as a hallmark of fibrosis because it specifically activates profibrotic genes, including collagen 1A1. So we were very interested to see whether this mechanism was activated in CAFs. So what we did was to inhibit the EP300 in our CAFs. And as you can see, when it's inhibited, we could easily decrease HS3K27 acetylation. So we use HS3K27 as a readout of this hyperacetylation. And when we did a proteomic analysis of EP300 inhibited CAFs, we found a strong decrease in production of most of the collagens. And you can also see indeed a strong decrease when in this microscopy analysis where we have CAFs in monoculture, and in red we visualize collagen 1. And we also found that this regulation occurs at gene expression levels because we found the collagen 1 and collagen 6 mRNA levels strongly decreased. So the interesting observation that came from this experiment is that we also found that the PYCR1 was strongly downregulated. Now, PYCR1 is the last enzyme that basically support protein synthesis from glutamine. So basically, this is the first link that we found between collagen production and protein synthesis. And I showed you before that collagens are highly rich in protein amino acids. So, but before investigating this further, we wanted to see whether these gene expression changes trigger by EP300 were regulated by acetyl-CoA levels. So to do that, we inhibited the production of acetyl-CoA in the cytosol by inhibiting the ATP citrate lyase in CAFs with an inhibitor. And this was sufficient to reduce histone acetylation, as well as collagen 1, collagen 6, and PYCR1 expression. And we could rescue this phenotype by treating cells with the supraphysiological levels of acetate. And acetate is basically an exogenous source of acetyl-CoA because when it enters the cells, is converted in acetyl-CoA. As you can see, indeed, we could rescue both histone acetylation and collagen and PYCR1 expression. And we then wanted to see whether this was dependent on PDK2. And what we did in this case was to overexpress in our CAFs PDK2. And this overexpression was sufficient to reduce histone acetylation, as well as collagen 1, collagen 6, and PYCR1 expression. And again, we could rescue this phenotype by providing the cells with exogenous cytosolic acetyl-CoA, giving supraphysiological levels of acetate. So now we know that this inhibition, so the dysregulation of the pyruvate dehydrogenase is responsible for a gene expression, a strong gene expression change in our CAFs. But is there a link between collagen synthesis, collagen expression, and PYCR1 levels? So what we did here was to take our normal fibroblast, silence PDK2 that I showed you before that are increasing levels of acetyl-CoA. And what you see here is that this is also sufficient to induce collagen expression. So we have increased the collagen levels, but importantly, when we inhibit PYCR1, these levels are significantly reduced, suggesting that there is a link between collagen production and protein synthesis. Now, when we did our metabolomic analysis of the different normal fibroblasts and CAFs, we actually also observed that our cancer-associated fibroblasts produce much more protein from glutamine compared to the normal fibroblasts. And we wanted to investigate in vivo whether this newly synthesized protein could actually be used to make collagen proteins. To do that, we transplanted our mice with MCF10DCIS.com breast cancer cells, let the tumor grow, and then we infused these mice with heavy label glutamine. And then we harvested blood and tumor for metabolomic analysis and the tumor for proteomic analysis. If we start with the metabolomic, this analysis told us that we could detect indeed heavy label glutamine both in the blood and in the tumor. We also found heavy label glutamate, so we know that glutamine is then metabolized into glutamate both in the blood and in the tumor. And importantly, we found that the glutamate was used to make proline, and we could detect heavy proline both in blood and tumor, and we found particularly interesting that it was actually more abundant in the tumor. But the most important result came from the proteomic analysis. So we analyzed the proteome of the tumor, and then we looked at the presence of heavy label proline in collagen proteins. And what you can see here is that we basically identify the presence of heavy labeled peptides in collagen 1 proteins. So this is the first proof of principle that indeed in vivo, the tumors are using circulating glutamine to make a proline for collagen synthesis. So we then wanted to see the function of PYCR1. First of all, as you can see here, PYCR1 is actually upregulated in our cancer-associated fibroblasts. And when we silence it with siRNA, shRNA, or inhibit it with an inhibitor, you can see that we significantly decrease the production of protein. And what this these images show you is that when we inhibit PYCR1, we strongly inhibit the production of collagen. And importantly, we can rescue the production of collagen only with the supraphysiological level of exogenous proline, but not with the physiological levels, really suggesting that the fibroblasts need to make their own proline to support the collagen production. And we know that this effect is due to the translation of collagens, this phenotype, because when we inhibit PYCR1, we don't see a decrease in mRNA levels of collagen. However, when we do a differential ribosome codon reading analysis, we can see that ribosome particularly stop at proline codons, and among the most affected proteins, so where the translation is reduced, we have several extracellular matrix proteins, including several collagens and collagen 1A1. So we then wanted to see what happens in more physiological conditions. So here we are inhibiting PYCR1 in a co-culture of CAF and cancer cells. We inhibit PYCR1 with an inhibitor. We reduce the collagen production and, importantly, also cancer cells proliferation. And we know that this can be due, at least in part, to collagen. In this case, we have a culture of CAFs, silence or not, for PYCR1 and normal cancer cells. We see that inhibiting PYCR1 in the CAFs reduces the proliferation of the cancer cells, and we can rescue this phenotype by providing soluble collagen to the co-culture. And finally, I want to show you what happens in vivo. So in this case, we did a co-transplantation models of breast cancer cells, patient-derived CAFs, control or silence for PYCR1. And reducing the expression of PYCR1 in the CAFs was sufficient to reduce tumor weight, tumor size, and tumor collagen measured by second harmonic generation. But we didn't see a difference in CAFs. And in a more aggressive tumor model, where we co-transplanted 41 cells with the CAFs, again, reducing the expression of PYCR1 in CAFs was sufficient to reduce tumor weight and tumor size. It also reduces tumor collagen as measured by serous red staining, but not the total amount of CAFs. We didn't see differences in blood vessels or hypoxia. However, we could see a very strong decrease in amount of circulating cancer cells and cancer cells that reached and colonized the lungs. So really suggesting that inhibiting PYCR1 in CAFs is sufficient to reduce metastasis. So just summarize the model. What I showed you is that we have identified two metabolic pathways that can regulate collagen production, one based on increased acetyl-CoA production and one based on the synthesis of proline amino acids. But importantly, what I have shown you is that we have identified a vulnerability to target extracellular matrix production, in particular by targeting PYCR1, because I show you that it's sufficient to reduce collagen production, inhibit cancer cell growth, and the formation of lung metastasis. So, and with this, I would like to thank all the people that have participated to this work. I would like to highlight a particular Emily Kay, who has done most of the work, all the amazing facilities that we have at Whitson, all our collaborators, and I wanted to mention that we are looking for one postdoc and one PhD student. If anyone is interested, please get in touch. And I wanted to thank you for your attention, and I'm happy to take any question.
Thank you very much, Sara, on a beautiful talk. I think we already have two questions. The first one, if I'm right, was Julie. So Julie, go ahead if you're there.
Yeah, I'm here. Thanks for the beautiful talk, Sara. I have a question about when you co-inject the MCF10.com with the fibroblast and you see a decrease in the tumor volume, do you see metastasis appear? I know it's a very non-invasive model, but do you see something like in the grow or go situation?
So we haven't checked that, also because we need to take these tumors very early. So basically after 2 weeks, we need to take them off because the human CAFs, they start to decrease in amounts. So we take the tumors when there are still some CAFs there. So therefore, I cannot answer, and that's why we moved to the 41 model.
Got it. Thank you.
So before giving the mic to Nita, why did you use the subcutaneous model with that cell line and not, because it's a classical model for intraductal grafting, right?
Correct. It's because in the original publication, they showed that there is not much difference in the phenotype of the tumors, whether you do orthotopic or intraductal and subcut. So I think we went subcut just because of that. But then with the 41 model, we went then fat pad transplantation. Yeah.
So the next question is from Nita.
Hi. Thank you, Sara. Great talk. I was wondering, I mean, it's really a very attractive thought that if we could interrupt the production of collagen, then we could inhibit invasion and metastasis. But collagen is such an essential thing physiologically. So I'm wondering if, you know, the translatability of this, whether this can be actually a valid therapy.
Mm-hmm. So I think that also what our data shows is that when you inhibit the endogenous production of proline, you are reducing collagen production, but you are not completely inhibiting the production of collagen. And this is because you have a lot of circulating proline, for example, in the blood. So it's one of the most abundant amino acids. So therefore, there is always some proline available. But basically, you are slowing down its translation. So I think for me that suggests that is indeed something translatable. And the other thing is that there are basically genomic, so you can have mutations for PYCR1, and the people who have mutations to PYCR1, they develop cutis laxa. So basically they have alterations in their skin, they become more wrinkly, but there are no, so it's not lethal, this disease, if you have mutations in PYCR1. If you have mutations in other protein synthesis enzymes, you can have a much stronger defects. So this would suggest that is compatible. And the other thing with life and with the potential treatment. And the other thing is that there are several reports that the PYCR1 knockout mice are actually non-lethal, and we are also working with these mice at the moment.
Okay, thank you.
Thank you. So we have one question from Tom, who is in the panelists, before two questions in the chat. Tom, still awake?
Yeah, I'm still here. Sara, absolutely fantastic. And as you say, this is such a complex area to be in. I'm just curious if you could sort of perhaps give some insight into your in vitro setup in terms of things like growth media. I mean, traditionally we put everything in DMEM, but I can imagine that would completely mask a lot of the subtleties in terms of metabolism of things. And on that, whether or not, for example, amino acid dropout might also help us to really get an idea of what's going on in terms of nutrient availability and that incorporation into sort of desmoplastic responses in tumors.
So a lot of questions. So the first thing that I can say related to this study is that we did all our experiments in DMEM, but then we repeated key experiments in DMEM with the proline in physiological concentrations and in physiological concentration of glucose and glutamine. Okay, and we could see a very similar phenotype. However, now we started working very extensively with the plasmax, which is a medium that contains the physiological levels of all the amino acids and other metabolites. And definitely we see that this has an impact in the metabolism of the cells. So we really want to start understanding a bit better how the metabolism changes in more physiological conditions. But I think it's also very important that we understand what is the availability of amino acids in the tumors and also other metabolites, because at the moment, even with this new medium that we are using, plasma, so we are mimicking what is in the plasma, so in physiological conditions, but we don't know exactly what we have in the tumor. So that's something that we really want to investigate. And I think that this is a very, for me, it's something very exciting, because understanding the availability of amino acids and, for example, also how diet can influence how much of them you have available, can also give you an idea whether you can maybe intervene therapeutically with the diet instead of with the drug, or maybe in combination to have stronger effects. And I don't know if I've answered all your questions.
Absolutely, and that was my follow-up was, have you any idea about nutrient availability in tumors? But you've covered that. I think it's a fascinating area.
Of the availability of
just nutrients inside tumors, but you touched on that.
Okay. So I think there are beautiful works from Matt van der Heijden, really showing the interstitial fluids in tumors, what is the content of different amino acids. And I think that the take-home message is that different tumors have different contents of nutrients available. And I suppose it can depend also on the stage of the tumor and potentially the treatment that the patients had. But I think that is something that needs to be really explored much more to understand how to tackle the metabolism properly.
Great, thank you.
Thanks a lot. So we have 8 minutes left and lots of questions. I go for one question in the chat from Sirio Dupont. Nice talk, Sarah. Do you think the effect of collagen 1 on cancer cell growth is an adhesive/mechanic one, or rather a metabolic one, cancer cells eating collagen?
Oh, interesting. That's an interesting hypothesis. I actually thought it was more related maybe to integrin activation, but frankly, we haven't tested this hypothesis, so we don't know yet what is exactly the mechanism. Okay. But thanks for the suggestion about the eating.
Then moving back to people having the mic on. Ruth, you want to ask your question?
Yeah, sure. Thanks, Sarah. That was a fascinating talk. I really enjoyed it. And following up perhaps on the discussion with Tom, do you think that in a different setting, such as in pancreatic cancer, where the metabolism might be different, but collagen is very general, the metabolism in different tumors can be different, would you see a similar effect? And then also within tumors, do you think in more hypoxic or less vascularized areas, you would see different effects of such nutrient availability?
I can't say very much, but in terms of experiments, but my guess is that probably if we will target PYCR1 in breast cancer or in pancreatic cancer, we might see differences in total, in the effect, because I think what is very important to understand is indeed the availability of amino acids and also what the cancer cells do, because it depends also what are they need, and it depends also of whether the cancer cells themselves are, for example, making proline for other cells. So, for example, it has been shown that by Sara Maria Feint that there is the proline pathway activated specifically in lung metastasis in breast cancer, which would suggest that probably this will have an impact of what happens to the proline metabolism in the CAFs in the metastatic sites. So I have to say that related to proline, these are questions that we are very interested in, but I don't have a clear answer at the moment.
Okay, thanks.
Thank you. So we'll go for another live question from Simon Schworer.
Hi, Sarah. Very beautiful talk and beautiful work. I really love this connection between the epigenome and metabolism and collagen synthesis. And what I've been always wondering is why do you think it's so specific for PYCR, the role of, the acetylation and the role in proline synthesis in your system? And because we see that also the other proline synthesis enzymes are upregulated, but that doesn't seem to be an effect on acetylation there. So do you have any comment on this?
So, so how to say that? So I don't think that, so if we start from the acetyl-CoA and from this epigenetic regulation, I think we have focused on the proteins that we found the most regulated, but there were other proteins regulated. So it might not be that specific, and this is something that we want to investigate further. So what are the other changes involved? And in terms of proline metabolism, well, first of all, I found very fascinating that there is this direct link collagen and proline. And by the way, PYCR1 and collagen 1A1, they are on the same chromosome. So I don't know if there might be also a link generally in how their expression is regulated. But what we also know is that there are other enzymes for the synthesis of proline that are very important for the synthesis of collagen. So it has been shown, for example, for the LDH 18A1. So it's not only PYCR1, and actually I think that PYCR1 may have even a bit milder effects compared to other enzymes of the proline synthesis pathway, that when you inhibit them, you are actually much strongly inhibiting also NADH and ATP levels. So it may have a much stronger effect in the metabolism of the cells.
Thank you.
So we still have lots of questions within the chat. I'll start with the first one from Christina Vieira Bardon. Hi, Sara, amazing talk. Do you have any clue of which signal triggers this CAF phenotype? And then there's a follow-up on that.
So we have some, you mean, I suppose that what you're asking is whether PDK2, the downregulation of PDK2, what is the trigger? And we don't have a clear answer yet, but we have some data showing that TGF-beta may have this effect. And actually, it has already been shown that there is a link between TGF-beta activation and TGF-beta stimulation and pyruvate dehydrogenase phosphorylation. So that could be the link. And there are also gene expression data of high-grade serous ovarian cancer published by Samuel Mock some time ago, where they show that when you treat ovarian fibroblasts with the TGF-beta, the levels of PDK2 are actually strongly downregulated. So that might be a mechanism.
Okay, and then she would like to know whether you think that it's a possibility that acetyl-CoA could be secreted by CAFs and support tumor cell growth besides the role.
Okay, interesting. So that's also something that we haven't thought. And we have never looked at extracellular acetyl-CoA, but that's also an interesting hypothesis.
Then the next question is from Aurora Villardi. Have you ever investigated the role of proline, glycine, proline fragments in collagen production or disruption and tumor responses?
In what sense?
No idea.
Try to read it again. So the role of
ever investigated the role of proline, glycine, proline fragments in collagen production / disruption and tumor responses?
No, I would say no. So we haven't for sure.
Okay.
I cannot comment more than that.
Then a question from Diana Nunez. Does this CAF phenotype also occur in human ascites of HGSC or only in the primary tumor?
So this is what we see in breast cancer tumors, okay? And for the high-grade serous ovarian cancer, I would recommend you to go to poster number 1 and then have a chat with Teresa.
Okay, we have one question from Viviana De Rosa. Sorry, I need to read it carefully. have you investigated whether the alteration of PDCase axis can affect some change in glucose metabolic phenotype that could be useful to collagen disruption?
We haven't. We have focused mostly on the TCA cycle. TCA cycle, so what is downstream? We never looked upstream, so I don't know.
Okay. And since we are within the break time, I'll go quickly over the last few sets of questions. Jennifer asked first, do you think that targeting the availability of amino acids for collagen production could also influence basement membrane or collagen 4 remodeling, and could this play a role in cancer progression?
Yeah, that's something that need to be considered. So it's something is very interesting. So as far as I'm aware of, so there is a paper published in Cell Metabolism quite a few years back, where they used the proline-free diet to feed mice and look at the impact on tumor growth, tumors with the high or low PYCR1 expression in the cancer cells. And they saw a big decrease in tumor growth when the cancer cells were dependent on exogenous proline. So I suppose then that the lack of proline in the diet didn't induce, defects that supported the formation of metastasis. But it's something that definitely we should look at. Yeah.
And we have 3 questions left. One from Didem Caracas. Some studies indicate that decreasing the collagen might increase metastasis, since collagen creates some kind of a physical barrier to tumor cells. And this person is wondering, what is your opinion about that?
Yeah. So I think, that's one of my not struggle, but I really would like to find a clear answer here. And at the moment, what I think is that it depends on the tumor type, because most of the studies where you see increased aggressiveness have been shown in PDAC models. And I think PDAC is very unique because the amount of stroma that you get in that models in mice is very unique, because you don't get that amount of fibroblast or stroma, for example, in breast cancer or ovarian cancers. So I think from my point of view, it depends on the tumor model. And maybe it depends also on when you are targeting collagen. But this is a very important question that needs to be solved, is when should we target collagen so that you inhibit tumor rather than promoting it?
Then a question from Lynn Assad. I'm not sure I get it fully, but in your in vitro experiments, do you think that in 2D collagen 1 coating with different glucose levels in the media would affect tumor cell response? I'm not sure I get it right.
Tumor cell, so what would be the rationale? So if you coat with the...
I'm not sure, maybe in terms of proliferation, I have no idea.
I'm not sure.
Okay. I suggest Lynn asks you directly maybe later. And then
maybe we can have a chat in the gather. Yes.
And we'll finish with a last question from an anonymous attendee, which might be a technical question, saying because it's quite easier to grow primary CAFs in culture. So this person is wondering, how could you maintain them in culture, especially because there's a high chance, they have a high chance to be activated and transformed into myofibroblasts or CAFs after a few passages.
Yeah. So basically what we do is that we have some immortalized lines. So we are immortalizing both the normal fibroblasts and the CAFs, so that we can keep them in culture for longer and they maintain at least a part of their phenotype. So we don't culture them forever, even if they are immortalized. So we culture them up to a certain number of passages, and then we just saw a different vial. But we always check, for example, alpha-smooth muscle actin to be sure that the normal fibroblasts don't get activated, and for any reason the CAFs are losing some of their markers.
Okay, cool. So there was one last question, but I think we need to move on since we already used the break time slot. So thanks a lot, Sara. And
Okay, thank you, everyone. Thanks to Jackie.